I will tell you about an idea that's been percolating in my lab for almost 20 years and has a longer history than that, as Steve alluded to. That it's an idea to use interfering particles and develop them into antivirals. And it's a counter dogmatic or counter approach that some of my colleagues, such as Harmeet Malik, have called a holy grail, partially in jest. And I want to take you through a little bit of the history that Steve alluded to. Okay, so this starts quite a while ago with, I think, with Preben von Magnus in the 1940s. He was a Danish microbiologist, actually discoverer of monkeypox, and von Magnus and colleagues were studying many viruses in tissue culture and saw this phenomenon. When they infected cells in culture, they saw a virus rapidly increase, and then basically these boom-bust cycles or oscillations where the virus would drop and then recover. And von Magnus hypothesized that this was due to an incomplete virus or a spontaneous mutant that arose that acted as a parasite and interfered with the wild type. When the wild type got to very high levels, it would knock it down. But when the wild type got to low levels, there'd be nothing left for this incomplete virus to parasitize, no raw materials. So the wild type virus would recover and this would repeat itself. And he called these incomplete viruses. Others called them von Magnus particles, probably in jest, because there was no evidence that von Magnus had, no direct evidence for these particles, some indirect biochemical evidence. But it really took 20 years for the first direct evidence to be found. And I think it was Adelin Hackett, who in the early 1960s was, he's a microscopist at NIH in Berkeley, studying VSV now. And VSV typically forms these stereotyped bullet-shaped particles of uniform length, shown here. And if you can see my cursor, hold on, let me try and do this. This is better. When Hackett looked at these von Magnus oscillating cultures over here, what she saw were these short stubby particles shown here at this arrow 1. And these are actually mutations in the capsid protein that form these short particles. Everyone pretty much forgot about Hackett, unfortunately, and what they remember is this very influential and beautiful paper from David Baltimore and Alice Wang that Steve alluded to, where they renamed these particles defective interfering particles and showed that they were across a broad range of viruses probably acting, so a general phenomenon. There was a lot of interest after this paper from David Baltimore's group, and it led to a huge amount of funding and research to try and convert these defective interfering particles into therapeutics. And if you look at the history of defective interfering particles, unfortunately, it looks a lot like, it mirrors the empirical phenomenon over here. So if you just look at publications and funding over time, there was a lot of interest in 1970 after the Baltimore paper. Probably irrational exuberance on the part of the field because the technology didn't exist to really create these therapeutic versions of interfering particles. And the field fell really flat for 30 years. There was essentially very little publication or patent or any sort of funding for defective interfering particles for a good 20 or 30 years. And then in the early noughts and 2010s, a series of mathematical models that we published got the attention of some funding agencies, and they put a large amount of funding into a program to fund about 24 labs around the world to really try and revisit this phenomenon. But for a good number of years, it was a backwater. And I'm going to tell you about this late 2010s, and that's where we come in. So here's the outline of the talk. First, I'll tell you about the theory of a TIP, a therapeutic interfering particle, why we renamed it from a DIP to a TIP. Then I will tell you about the technology that we developed to try and find and develop these TIPs. I'll show you proof of this concept for HIV and maybe a little for Zika. This is still in review, even though we worked on it for many, many years. And really the main event I want to tell you about is the SARS-CoV-2 TIP. Show you the mechanism and the hamster efficacy. And this was published last year. We think it's the first evidence of a therapeutic interfering particle. And I want to warn you, I tend to give long introductions. So 10 minutes from now, if you still haven't seen a piece of data, don't be alarmed. It will come. I'm going to start off, as I always do, with the acknowledgements. Many people over the past 20 or more years in my lab and my group helped develop this concept. The hero here is Sonali Shettiverdi, who was a postdoc in my lab, now an instructor. She's a one-person lab, a force of nature, really amazing individual. I also want to shout out Davey Smith, who's our clinical trialist, and Rob Roddick, who co-founded a company with me to develop the technology and is the IND sponsor for the clinical trials. And this is just a more recent version of the lab, everyone masked, and celebrating Sonali's success. She's also the social coordinator of the lab, and she's going on the job market next year. Okay, so I'm going to start off with the problem. This is a well-known issue now. A paper from earlier this month in the New England Journal showing the efficacy of vaccines against viral variants of COVID, of SARS-CoV-2, both waning immunity as well as after the boosters, just the reduced efficacy to the variants, in this case, Omicron showing reduced efficacy relative to Delta. And for many people who study seasonal influenza, this is not a huge surprise. We have lots of evidence over the past few decades of influenza vaccines having reduced efficacy year to year due to drift and in some cases shift. You can see in some years higher efficacy and in other years lower efficacy. This is data from the CDC. This is a well-known problem that viruses will drift or mutate, vaccines do not, and we have to continually update or guess. A second unmet need or problem that we deal with in the case of both vaccines and antivirals falls under the general rubric of deployment. It's both access and adherence for antivirals, at least. And I think the HIV case really highlights this. We have exceptionally effective and safe drugs, antiretroviral cocktails for HIV, but getting these drugs and maintaining regimens for patients is incredibly challenging. And despite these continent-wide deployment campaigns, PEPFAR, for example, in the last year that there's good data available, 2019, there were over 1.5 million new infections of HIV. And that's despite these amazing drugs. So we know we have these two general problems. And the question we had was, can we in any way reach back to that old phenomenon, that old idea of defective interfering particles, and maybe use those to address these two problems? And so here's the concept. And I'm going to show it in the context of HIV, because that's where we originally had the idea. And then I'll move over. A retrovirus single strand, it's flanked, it's about 10 kb, 9 genes, and a single promoter in the 5' long terminal repeat. It's flanked by these two LTRs, and the 5' is the promoter. It's a very weak promoter, but one of the viral genes, TAT, feeds back and transactivates this promoter to make it a very strong promoter. We worked extensively on this feedback loop. So to make a long story short, when TAT is present, and I'm showing you the bottom half of the slide here is the HIV life cycle in half a slide. When TAT is present, virus is transcribed at high levels, all the genes are expressed, these RNAs are exported to the cytoplasm. These squiggly RNAs translate the proteins in purple, and if these RNAs carry the packaging signal, if it's not spliced out, they are efficiently packaged within the viral capsid, encapsulated, enveloped, and viral mature viral particles are emitted from the cell. So the idea was, if there was a defective interfering particle for HIV, and HIV was actually the exception in virology, there was no lentiviral or HIV defective interfering particle, bona fide one, that had been reported. But we hypothesized that if there had been a defective interfering particle, if there was one, it would look very much like the VSV or the influenza defective particles that von Magnus described. It would have a mutation in one of the genes, for instance, the capsid gene, GAG. This virus would not be able to replicate on its own. It would require the wild type virus in trans to help it. And our hypothesis, what we basically said was, let's take this idea to the extreme. Let's cut out all of the viral genes, all the trans elements, and what we'd be left with is this small piece of RNA that carried only the cis elements, in blue. So it had the promoter, it would have the packaging signal and all other cis elements. And we called this a therapeutic interfering particle, and I'll show you why in the next slide, why we named it. If we could produce these, they wouldn't do anything on their own, but they'd be transactivated by the viral proteins, TAT, for example. And if we could produce this and introduce it to a cell through standard genetic engineering approaches, for example, it would transcribe RNA in the presence of HIV. These RNAs, if we made them properly, would be shorter, be transcribed at a higher level because they wouldn't be spliced, for example. And because they all carry the packaging signal, they could stoichiometrically outcompete HIV for its own capsid and envelope. And what would be coming out of the cell instead of HIV would be these viral-like particles that would be phenotypically identical to HIV, but genotypically, they would carry the therapeutic interfering RNA. So this was the idea. It would be therapeutic because it would reduce the viral load. And if it reduced the viral load, it could reduce transmission. So there are many benefits to this concept that we explored theoretically in the computer for many years because we weren't able to build these. This was all really imagination at this point. And let me tell you why we renamed it to a therapeutic interfering particle. So here's the theory in one slide, schematically, no math. HIV, or the wild type virus, in this case HIV, is shown in red. In blue is the DIP or the TIP. Classic von Magnus dips need to have two criteria or two requisites. One, they have to conditionally replicate, of course. So if the blue dip gets into a cell, it can't do anything until the cell is super infected by the wild type virus, in this case HIV. Then it can replicate. That's criteria one. Criteria two is interference. The wild type virus infects a cell, produces a burst size of virus, a certain amount of infectious virus. In the presence of the interfering particle, that burst size is reduced. So those are the two criteria. And we added a third, a quantitative criteria, which is the concept of R naught greater than 1. So I used to have to explain what R naught was, but now everyone's an armchair epidemiologist. Everyone knows what R naught is. In this case, R naught is defined at the cell culture tissue level. A single infected cell, the number of new transduced or DIP transduced cells it produces. We said it had to be greater than 1. This would ensure stability of the tip and allow it to be therapeutic. So all tips are dips, not all dips are tips. And it turned out to be incredibly challenging to build a tip that had an R naught greater than 1. So I will not take you through the 20 years of failure to do this. It's a beautiful story, I think. I will just take you through one example. We had to build a screening technology, finally, to find HIV dips. I'm just going to summarize this in one slide. Basically, what we're able to do is build a high-throughput library. So this is a technique we call random deletion library sequencing, or RandleSeq. And this is a library of hundreds of thousands of viral deletion mutants, randomly positioned mutants of variable size. This library, then, we can use to screen. Here's a subset of the mutants just shown. All these mutants are bar-coded, these deletions. And so we had hundreds of thousands. Here's a subset of 20,000 or so. And we were able to screen this library for tips. So I'm going to show you, this is all published. I'm going to show you one example of this tip working. So what we did is put a blue fluorescent protein inside of the tip and transduce cells, and then put a red fluorescent protein in the Neph reading frame of HIV and superinfect the cells. And so I'll show you a 40-hour movie of this occurring, microscopy movie. First, the cells were transduced. Nothing happens because HIV is not present. And then when we superinfect the cells, I'm going to show you what happens. This is the beginning of the movie, the first 15 hours. Now I'm stopping the movie at 15 hours. And you can see a little bit of red coming up from HIV, tiny little bit of blue, but not much. And I'm let the movie go forward. Now the tip is transactivated, takes over, expressed at very high levels. And you can take the supernatant off of this culture and throw it onto a new culture. We did this in a bioreactor setting, so to extend it. HIV infected cells on the y-axis, and you can see HIV really destroys the culture in about 3 weeks. Because of our reactor setting, we're able to extend it out to about 3 weeks. And in the presence of TIP, a single administration of the TIP at early time, we're able to extend that culture and protect the cells pretty much as long as we can hold the reactor, in this case, 45 days. We were able to build tips for SIV. This is now unpublished work I'm showing you together with Nancy Hagewood. Reduce viral set point in rhesus macaques from a single administration of tips. So single administration of tips dramatically reduced viral set point in SIV. I'm going to go through this very, very quickly just to give you a taste. We found same reduction of viral set point in humanized mice. This is the BLT model, about a 1.5 log reduction in viral set point from a single addition of tips sustained over many months. And in patient cells from a cohort of patients that maintained by Stephanie Stratti at UCSD, these are injection drug users, we're also able to see reduction in viral expression. Okay, so that's just the proof of concept that we can add. These things are buildable. Now I want to tell you about the SARS-CoV-2 tip, and this is all work by Sonali, who really in April 2020 went into the BSL-3, hasn't come out since, and she spearheaded this. She's the co-corresponding author on this paper. I've never done that with a postdoc before. She's really the driving force behind this and an incredible individual. Okay, so here's what Sonali did. Very much like the HIV case and using some of the Randall-seq data we had, here's the SARS-CoV-2 genome, just for comparison. It's flanked by 5' and 3' UTRs, and it's much larger. It's a 29 kb. The genes are shown here. What Sonali did is really take the 5' and 3' UTRs, shown in blue over here, and a intervening IRS-M cherry reporter in between was stuck in. She built RNAs, made two different tips here, I was showing you. Made RNAs, 5' capped them, poly-A tailed them. They were about 2 kb, so much smaller, less than 10% of the genome. She also built a second version, which is a little larger, of 3' and 5' UTR. And a control RNA, which is size matched. And we built many different control RNAs. I'm going to show you one of them. It's also 5' capped and poly-A tailed. This one carried a luciferase in place of the 5' or 3' UTR. And then she did really classical virus yield reduction assays where the cells were transfected with these RNAs, these are Vero cells in culture, and then superinfected with SARS-CoV-2 and collected culture at 24, 48, 72 hours and assayed for viral yield reduction. So here is RT-PCR data for spike protein or E or M, it doesn't really matter. So sorry, spike RNA or E or M compared for the control RNA versus tips, tip 1 and tip 2. And there was a very sustained 1.5 to 2 log reduction in viremia, log 10 reduction, very significant. And this was at all time points. I'm showing you 48 hours, but it's at all time points. And if you looked at infectious virus by PFU, platforming units, same thing, about a 1.5-fold reduction, a 1.5 log reduction, excuse me. So one of the first questions was, are we just inducing an innate antiviral response in these cells because we're transducing or transfecting large amounts of RNA? It's unlikely because the control RNA didn't produce that. And these are 5' capped poly-A tailed RNAs. And if you read the original papers from the Weissman lab, it turns out that 5' capping, poly-A tailing does the same thing as polyhydrodenylate. You don't really see an antiviral response, but we tested it anyway. So we looked at pro-inflammatory cytokines by RT-PCR in these cultures, TNF, IL-6, interferon. There might have been a little bit of an increase, it looked like, in interferon. It was not significant, but just in case, we looked at downstream interferon stimulated genes, ISGs, and there was no increase in those genes. So it looked like this is just an artifact of the culture, maybe. And if you looked at the downstream or the upstream, RNA sensing genes, TLRs or RIG-I, there was also no increase in the innate responses. So really consistent with the theory that 5' cap poly-A tailed RNAs should not be inducing innate antiviral immunity. And I'll show you some similar data from hamsters. So the next thing that was important is we needed to have an R0 greater than 1, otherwise it's not really a tip, right? So because of the IRS-M cherry, Sonali was able to collect supernatant and assay it, the supernatant by a supernatant transfer on naive cells. This is flow cytometry data showing efficient M cherry in the naive target cells. You can calculate an R0, it's greater than 1. I won't take you through that. Instead, I'll show you a very elegant experiment that she did, which is a long-term culture where at 1 day pre-infection, she transfected the cells and then carried the culture by supernatant transfer out to 3 weeks. And the tip reduction in viral load is sustained, 2 log reduction in viral load is sustained for 3 weeks, even though there's a single addition of the tip. And if you look at the last time point over here, at 3 weeks, you see a substantially increased TIP level. And really, that only could have occurred, right, if the R naught is greater than 1 and the TIP is carried. Okay. She did some really beautiful and elegant reconstitution assays. This is actually her specialty. So in this reconstitution assay, the cells are co-transfected with plasmids for the trans elements, the spike, envelope, matrix, and nucleocapsid, and the RNA, in this case, either TIP1 or a control RNA. And then VLPs, if they're created, are collected and assayed on target cells. And you can see here, in the only case where you saw a functional VLP in the target cells was in the TIP case. The control RNAs did not produce functional VLPs that transferred. She also did really beautiful EMSAs where pull downs of different SARS-CoV-2 proteins, either RdRp, N, envelope, spike, and only RdRp and nucleocapsid really co-precipitated with the TIP1 RNA and specifically co-precipitated, arguing that the TIP RNAs are actually stealing, stoichiometrically stealing RdRp and N protein. More work to do here, of course, but this is the first. So then the question was, are these TIPs effective in vivo? Are they therapeutic? Because we can show a lot in cell culture, but do they have any effect in an animal? So the first problem was, how do we actually deliver these to an animal? RNA, you can't nucleoffect or transfect an animal. And so we settled on these lipid nanoparticles, which are the same technology that Pfizer and Moderna use for their RNA vaccines. This is the exact same lipid mixture that's used by those two companies, 4-lipid mixture with a pegylated lipid, an ionizable cationic lipid, a helper, and cholesterol, and the TIP RNA. And you make these lipid nanoparticles that are about 40 to 80 nanometers in diameter. We delivered these intranasally, first to a mouse using a luciferase control RNA. This is just the RNA alone, a naked RNA. You can see a little bit of luciferase expression in this mouse, but if you give the luciferase RNA in an LNP, really dramatic delivery and expression, even into the lower respiratory tract, not just the upper respiratory tract. So we tested this in a TIP RNA setting in the humanized K18 transgenic humanase 2 mouse, and we saw pretty dramatic protection against weight loss. So this is a pilot experiment. The control RNA treated mice really went pretty low. We almost had to stack them, and the TIP treated mice maintained weight. And so we moved to a more physiological model, the Syrian Golden Hamster. And this is work done together with Tom Rogers and Davey Smith at UCSD. The Syrian Golden Hamsters were challenged, in this case, with a very high viral load, 10 to the 6 PFU. And this first experiment that I'm showing you, intranasal delivery of the TIP LNPs, this was a prophylactic experiment. So the TIPs were given 6 hours prior to challenge. So first experiment, I'll show you post-exposure in a minute. Animals are carried out to day 5. Day 5 is the standard for looking at peak viremia in the lungs. And so that's what we did. And a very, very dramatic, about 2 log reduction in viral load in these animals in the lungs, infectious viral load. This was also by RT-PCR and other measures. You see that sometimes. It might have been a delivery or administration issue. These animals also were protected against severe disease. So this is histopathology of the animal lung. The control RNA treated animals with TIP RNA treated animals, and you can see a very characteristic pulmonary edema in the alveolar sacs filled with fluid, shown by the astrocies over here, whereas the TIP treated animals are quite clear. You can quantify that and score it, and it's a highly significant protection against pulmonary edema, as well as inflammation. We did RNA-seq on those lungs, and the TIP-treated animals looked much better than the infected animals. Here are 3 inflammatory cytokines, pro-inflammatory cytokines, pretty significant reduction in IL-6, and the TIPs themselves did not induce an increase. This is a lot of data. I'm just going to, for the interest of time, skip it. And I'm almost done. So the question is, could TIPs be delivered post-infection? So we did a 12-hour post-infection delivery. This is pretty much the last time point you can give an intervention, because after these animals recover, and after about 12 to 15 hours, they begin to develop their adaptive immune response. So you can't really demarcate whether the recovery of the animals is due to their antibody response or due to your intervention. So 12 hours is the time we went in. It's also the time other people go in with malnupiravir and other antivirals. Pretty dramatic effect, again, highly significant effect on reduction in infectious virus, and the animals' lungs were protected against severe disease again. We also were able to find a strong robustness to variants. The IC50 really did not change as we went from the archival Washington strain to the beta variant. This is a very similar IC50 to malnupiravir, in cell culture. It's around 300 for malnupiravir, which is about 100 nanomolar. And some evidence, very preliminary, that this might actually be robust even out to the 2003 Hong Kong SARS-CoV-2 virus. And then I want to show you one piece of unpublished data in the last slide. It's now with the Delta variant. This is a co-transmission experiment to see if TIPs could reduce transmission of viruses. We know vaccines have a sort of mixed record of reducing viral transmission, especially for the variants. So this is a sort of standard reported approach where a Syrian Golden Hamster in gold here, source animal, is infected and then post-infection treated with TIPs or control RNA, single administration, and then co-housed with a contact animal in silver. The co-housing is for 8 hours. It's a very efficient transmission, even with this 8-hour co-caging. Animals are split and then harvested on day 5 or 6. Lungs are harvested. And here's the data. This is the nasal shedding from nasal washes of the animal. This is RT-PCR. Actually, sorry, this is infectious virus by PFU. You can see that there's reduced nasal shedding in the TIP-treated animals. And in fact, by day 4 and day 5, it's below detection limits, which is pretty dramatic for us. And if you look at the animal's lungs, really quite remarkable reduction in viral titer by PFU in the source animal lungs, and it's also in the contact animal lungs. They still get infected, but with a highly reduced amount of virus in their lungs 5 days later. And I'm happy to tell you about the reason that there's increased viral load in the contact animals compared to the source animals. It's a technical point that people have seen before. So that's really it. I just want to remind you, summarize. I told you about this concept of TIPs. Gave you some proof of concept data from HIV in rhesus macaques and humanized mice, and then showed you what we think is the first, at least published TIP, which is the SARS-CoV-2 TIP. The HIV TIP is still somewhere in the reviewer ether. The SARS-CoV-2 TIP is a single administration, intranasal administration, seems to prevent severe disease. It seems to be robust to mutational variants as far as we can tell. We think uniquely reduces viral transmission, where we haven't seen that with other approaches so far. And we do have a phase 1 trial funded that is, and studies are underway to enable that phase 1 trial. So I'll leave it there and take any questions. Thanks again.
Okay, Leor, thank you. So I have one question. If you envisioned using this therapeutically, and maybe you're not, but if you did, how long would you envision the protection would last after administration?
Yeah, so there are 2 ways I think to think about it. Is in the prophylactic sense, pre-exposure prophylaxis sense, and I think RNAs degrade quite rapidly. There are ways to improve that mechanically with lipid nanoparticles in different formulations, like slow release. So far, we've been able to do sort of a 48-hour pre-exposure prophylaxis. So you'd have to be taking these nasal sprays, in the same way that you take Tamiflu, for example, pre-exposure prophylaxis, which is the evidence shows that's the best use of Tamiflu, right? In a household at-risk setting. So that would be one way to think about it. Post-exposure, I think it's very hard to treat late in infection. there's very little evidence that any intervention can do anything after day 5, day 6. So I think we'd be in the same, this concept would be in the same time frame. It would be the first, 5 to 6 days. That's where 12 hours in hamsters seems to sort of roughly translate to about the day 5 in humans. That's where you can give monoclonal antibodies in hamsters. That's where you can give Paxlovid in hamsters. And after that, it becomes very hard to do anything. So I think we're in the same time range.
Yeah, no.
Question?
Every intervention has to be ASAP, basically. Yeah, right.
We do have some ideas that this might, this is, I'm really going to speculate here, but, that TIPS could sort of turn the wild type virus infection into something like a, live attenuated infection where you're reducing viremia, but you might get a long-term immune response afterwards for the variants, for example, but that's still to be tested.
So Eric, your hand is up.
Yeah, hi Leor. That was a fantastic talk. I have what is a terrible, naive question. Steve's going to laugh at me.
This is the best.
Why isn't it? Why don't the SARS-CoV-2 subgenomic RNAs act as natural tips?
Yeah, it's a great question. Maybe the best way to answer it is with some data. I can speculate, but let me. Okay, here. Let me show you some of the control RNAs that we tried, and this might give you an idea. I don't know if it's so. And this is what the reviewers asked for. So they needed all these control RNAs. One of them was a 5' only. So this is 5' only IRAS M cherry, and this one is IRAS M cherry 3' only, which is this one is sort of similar to the subgenomics, right? And then this one is an M matrix RNA with stop codons because we don't want to produce actual matrix protein. And here's a spike RNA with, again, stop codons, so we don't want to produce a spike. You can see none of these really produce an interference effect. And so why is that? I think because the 5' is missing the 3', so it doesn't package efficiently, it doesn't steal efficiently. That's our speculation. We have a lot more mechanistic work to do, molecular mechanism to figure that out exactly. Partially.
Thank you.
Yeah, I was going to say, presumably they don't compete, right? Yeah.
Yeah, I think cis-trans, it's all about what are the requisite cis elements, and
And so then the question, as it always was for HIV, is what would be acceptable to the world in terms of taking something that's a subgenomic fragment of something pathogenic. You have to persuade people that this is acceptable.
Yeah, you're absolutely right. This is, I've been campaigning and it's, we've made it part of the way. I think the monkey data for HIV helped, especially with the FDA. But I think there's a long way to go. Even with regulatory agencies, we're going to have to do a lot to convince them. But if you have any ideas, I would love to hear it. It's I think it's going to be a career.
Well, the good news is that it's not going to revert to give you live virus like the Neph minus Ronde-Rosier defective virus that was a concern that in fact could give live virus. So yeah.
Right. I agree that it's the concerns are circumscribed compared to that.